going to talk briefly about how we brought cures to our students at Johnson County Community College. We began in the early days of 2014 implementing the Tiny Earth curriculum with our students, and I want to talk a little bit about how we do that and how that project has grown into an interdisciplinary team that brings cures to our students in multiple classes. First off, we chose Tiny Earth as our model for a cure because it brings a lot of relevance to our students. We first introduced this cure into our microbiology courses, and this is a course that is designed for pre-allied health students, those students that are going into nursing, dental hygiene, pre-pharmacy, and so these are students that are typically not going to end up doing any research in their continued education and may not have had any experience in research coming into this course. We also like Tiny Earth because it really focused on finding new and novel antibiotics, and that is something that is incredibly relevant to our students that are going into nursing fields or pre-pharmacy fields, and it's something that's easily students can easily see the benefit and value of this particular project. So Tiny Earth, you can see the word cloud here. It's based out of the University of Wisconsin-Madison, and we partner with them to bring curriculum and the sort of the network of both faculty and students into our classrooms. Tiny Earth is spread out all over the globe, so you can see here that the dots represent institutions. Our dot is somewhere right there in the middle here in Kansas City, and you can see a picture from this symposium, the last in-person symposium that we held, just showing you the faculty that are involved in this effort across the globe. So the Tiny Earth curriculum begins with students finding environmental samples, typically soil. We know that bacteria in the soil produce many of the most useful antibiotics historically, and so it's a great place to go looking for new antibiotics, these chemicals that are going to kill bacteria and potentially pathogens. So students have the ownership of finding the soil sample that they most care about. So many times students will bring soil in from their backyard, their garden, their family farm, a particular place that is meaningful to them, and they're very excited and eager to find out, does my soil have any antibiotic producing bacteria? So after we take the soil out they bring their soil samples in, we then do some dilution, so there's a great quantitative aspect to that, and they plate those dilutions onto agar plates. This allows us to teach our micro students about colony diversity, the difference between different types of bacteria, colony morphology, kind of seeing the different kinds, and then also recognizing and looking very closely at these plates to identify what we call potential candidates, that is, bacteria that are producing an antibiotic. You can see here on this plate on the right that the zone of inhibition, this particular colony of bacteria is producing an antibiotic that is inhibiting the growth of all of its neighbors. So this would be an excellent candidate to choose for that student. So they have the opportunity to find and look at their plates from their soil sample to find these bacteria. We then make sort of a collection. So you can see here there's 7 different bacteria that a student has collected. Here's another plate showing the kinds of diversity that we see in these bacteria. So students very carefully transfer these bacteria from their original dilution plates onto what we call these master plates or these collection plates. And that again is skill building for our students and the chance for them to sort of curate this collection of potential bacteria that they can then do some screening on. We screen these bacteria, so we match them up against what we call the escape relatives. These are bacteria that the health industry and our medical industry really needs an antibiotic for. So these are particularly difficult to treat bacteria, and it's an acronym for 6 different types of bacteria. So we use safe relatives in our lab because our students are freshmen or sophomores and don't have a lot of BSL-2 experience, so we keep escape relatives in the lab for them to work with. You can see here that there's lots of different ways that we match these bacteria up, and this is one of those opportunities for students to be creative and to be innovative in figuring it out. You want to get the bacteria that they have found, their potential candidate, as close to the escape relative as possible, but to make sure that we both are able to grow. So in some cases we'll grow the escape relative, the bacteria we're trying to kill, in a lawn on the plate, and then put little patches of the potential candidates on top of that plate. So you can see here that this particular strain, SMR2, is creating a zone that's killing off Staph epidermidis. We've also seen students creating sort of different kinds of lines or patterns on their plate where they'll streak in one direction for the escape relative and the other in one of their chosen candidates. This one, I love the name, is Big Blob, right? So you can see that this particular bacteria is inhibiting E. coli. So there's an opportunity for creativeness and innovation in how the students set up these plates to determine if one of their bacteria create this kind of zone or is making an antibiotic. After students do that screening, they then pick a single candidate. So maybe they've found a couple candidates, maybe the student next to them screened all of the candidates from their soil sample and didn't find any that inhibited the escape relative. So we do oftentimes have sort of an adoption day where bacteria, students that have more than one bacteria can lend out their bacteria to another student. We then go through a lot of the microbiology curriculum in identifying and characterizing these bacteria. So we do staining, we do metabolic testing, we test to see whether they grow in aerobic or anaerobic conditions. So you can see some examples of those metabolic tests here. We also get a chance to send these bacteria out for 16S gene sequencing, and that allows our students to have exposure to what genotyping looks like, how to analyze sequence data. They have the ability to load a gel and do PCR, so they get to see that molecular approach as well as the more traditional microbiology approach of using the metabolic tests. So they get to identify the bacteria that they have found in their own soil sample and note which types of antibiotics or what types of bacteria it might be producing. At the end of this, we oftentimes have bacteria that fall into a number of different categories. So we've had 200 total bacterial candidates that have been found by students. 77 of those bacterial candidates seem to be unique, meaning that we don't, if we have unique different strains. So if they've been sequenced, they are not the same as anything else that we have in our current collection. So we're maintaining about 77 of these strains. You can see here the kinds of organisms that are inhibited. Interestingly, in orange over here, we have about 10% of our bacteria in our collection inhibit both a gram-positive and a gram-negative. So they're producing something that is broad spectrum, or they're producing multiple compounds. You can see that we oftentimes, the highest percentage of our candidates inhibit staph, but we also see a number that inhibit other types of bacteria. Okay, so what the end result is students end up at the end of the semester with an excitement. They have their research project. Many of them then choose to do additional research with us, and that's pretty unheard of at a community college to get that much excitement. So we have a number of students that have done independent study in an additional semester. Here's two of those students here presenting their work at national and regional conferences. We have some students that decide to stay on and do, as part of an honors program, a research experience. And then I want to talk just briefly about students that either go on or have an additional opportunity to use Chem 140 as their research experience. Here's an example, again, of some of that independent research students. So this particular student found a bacteria that inhibited the soil and did a lot of work to optimize the conditions to grow that bacteria and produce the most antibiotic possible. So that's another opportunity for these independent research students. Many of the students that work with me end up doing a sort of side project that I've been developing on endophytic bacteria in seeds. So here you can see just different seeds types that we've looked at and how many bacteria we've had. And then in green is how many of the strains that we found actually inhibit other escape relatives. And so I've had a number of students working on this particular project, and it's been a really great sort of second semester addition to our standard sort of tiny earth curriculum. Okay, so we wanted to get our chemists involved in this work because many times our students get to the end of our semester and they have identified a bacteria. They know that it inhibits some of these escape relatives. They've gotten some genetic data on it, but we haven't been able to actually work with or isolate the organic compound that it's making. So in Chem 140, which is a non-majors organic and biochemistry course, my colleague Melanie Harvey takes the bacteria from my micro students. We choose sort of the top 5 to 10 bacteria for her students to then work with. So we grow large quantities of this bacteria up. You can see here they chop up the bacteria if it's growing in solid media. They add an organic solvent. Oftentimes this is either methanol or ethyl acetate, and they freeze the bacteria. They then extract the organic layers and the aqueous layers to run tests on. Some of the tests that we've been able to do with these students is different types of chromatography. So here's thin layer chromatography. We've also done some column chromatography. We also can use those thin layer chromatography plates to do an agar overlay, so we can see which of the spots on the TLC plate is actually containing the compound that inhibits the bacteria. So here you can see a TLC plate in the bottom of this agar dish. Staph epidermidis was plated in a pour agar, so the bacteria was mixed with the agar and then poured over in this plate. And you can see this little zone right here is the compound that's inhibiting the Staph aureus that's made by that bacteria. So students in this chemistry course get opportunities to learn about different separation techniques, and they get to further the research that was being done in the microlab by really isolating what compound it might be. We're also working on sort of spot playing, sort of directly adding these extracts to the top of the agar. We also have a tool that can help us embed the extracts in the agar to help better deal with things like drops running together, as you can see here. So of course there's always challenges, and our students are really excited to jump right in and help us solve those challenges when we need to. All right, at the end of the semester for both microbiology and for chemistry, we have a CURE Symposium. So the faculty that's involved in this project has worked hard to create an opportunity for our students to present their work. So in the fall semesters, they use the CURE Symposium, which is really focused on students doing cures at Johnson County Community College. And in the spring semester, there's a more general math and science poster symposium that's put on by the Math and Science Division, and our students are able to present at that symposium. So here's a nice picture of what that looks like. We tend to have hundreds of students presenting, so we'll have multiple rounds of student poster presentations for them to get. And it's very festive. We usually have sponsor tables and food, and we oftentimes get both our president and our board of trustees members to show up to these symposiums, which is fantastic to make our students really feel valued. Some fun pictures from these symposiums. So you can see here some students presenting their work in both chemistry and in micro. And here's a board of trustee member coming in and talking to 4 different students about the value of what they're doing and how excited and enthusiastic he is to hear about their research. All right, so just to thank the Math and Science Symposium folks, Lori and Brenda, who do a lot of work on our campus in putting that spring symposium on every single year. Melanie Harvey's been doing some educational research that aligns with her Chem 140 students, and that's been funded by NSF. We have all of my student researchers have been incredibly important in creating this data and helping this program be successful. Of course, Tiny Earth, and 2 of our sponsors for our symposium, our Collaboration Lab, as well as Norton Publishing.